From the Cosgun Lab and the Bioengineering Media Lab at Georgia Tech, welcome to Lab Coat Conversations, where you too can join the discussion on the next academic breakthroughs. Join your host, Hector Torres, as we talk to Georgia Tech researchers. This episode, please welcome Karmella Haynes. So we're going to start off with, can you tell me just like a little bit about yourself, who you are, what you do, general areas of interest in your research, things like that?
All right. Well, so first, thanks for inviting me. I'm Dr. Karmella Haynes, and my general interest is in synthetic biology. And that interest started when I was just finishing my PhD at Washington University in St. Louis, where I was doing what looked a lot more like traditional biology research, right? Sort of developing hypotheses based on what people knew so far about how genes are regulated. So gene regulation's always been a really basic interest for me. Since I learned about DNA in high school, the next big question I had was, well, if all the DNA is the same in all of your cells, how do cells choose which ones to turn on and off? And that question had always fascinated me, how a cell can just take like a general blueprint and then, all the mechanisms that help the cells decide what to do with all of that information. So then as I was finishing my PhD, I had learned a lot about, the proteins involved in helping those cells make those decisions, but then started asking bigger questions like, okay, well, can we actually control it? Can we get a more precise quantitative understanding of how all of that stuff works? And also, I got curious about how we were integrating, this rapidly developing field into education so that students would be prepared to, become scientists themselves or even enter the job market, given the pace, right? The fast pace of bioengineering and things that had to do with quantitative biology. So I was a postdoc at Davidson College for 2 years as a teaching research fellow. So it was half teaching, half research. And then that was the first time I had encountered synthetic biology. And I kind of fell in love with it because it was it was a paradigm for understanding biology in a way that I just was not taught, right, or aware of during my PhD. And I liked this idea of taking basic concepts from machine engineering and then using it as a language for understanding biology. And I just love that. I thought that was super creative and groundbreaking, and it lets you design types of experiments that you wouldn't as a more traditional biologist would. I decided to do another postdoc after Davidson, so that one was really short, and while it was a fantastic experience with learning how to teach at the cutting edge of science, I felt like I needed some more time to develop my own projects and concepts and start to actually work on building a research program. So I went to Harvard Medical School and did a postdoc for 3 years with Pam Silver in her lab in the systems biology department. And that was quite a time. I really enjoyed that a lot. It was a ton of fun. And I really felt like I had a lot of room to explore. And then so that actually helped me to develop the foundation of our current work. So I had my first faculty position after that was actually at Arizona State University in Tempe, which is near Phoenix, Arizona. And so I built a small research team there and also stayed involved in undergraduate education, very active in that. And then so that's where I had developed our current platform where we take proteins that reside mostly in the nucleus and are busy sort of helping to organize the genome and turn genes on and off and regulate entire gene expression programs. And so as a starting point, we started looking at that in cancer, and then we started getting a little bit more focused on breast cancer. And we're able to, publish some really good papers. So then I felt, that I could, there was things to explore that were a little bit beyond what we had been working on. So then I decided to move to this program, right, biomedical engineering, because of all of the opportunities to collaborate with folks who were closer to translation, right? So right now I have a research lab that's right across the street from the Winship Cancer Center. And there's all sorts of really cool disease research happening in this environment. So I just sort of took the opportunity, moved here, and then the rest is history. So I've rebuilt a brand new research team since then. So I moved in 2019, and now I have 4 PhD students and 2 undergrads, and we're currently recruiting a postdoc. And that's just yeah, my intro, bit about me.
Yeah, that's a lot. Very experienced. Thank you. So you mentioned a little bit about your research, and of course, I researched you ahead of time. Um, and your website mentioned something about PCTFs. So I just wanted to see if you had any more information on that, and if you've seen this type of technology have benefits in like the greater society as well.
All right, absolutely. So happy to talk about that. So PCTFs. So the PCTF is an acronym, right? Because scientists and creators are always busy coming up with acronyms. So the it's short for polycomb based transcription factor, right? So PC is a shorthand name for polycomb, which is a protein that's very familiar to folks that study epigenetics. So this whole, business of, how cells decide to switch genes on and off. And then in addition to that, how, once those gene expression states are set, it's very important for all of the daughter cells in a population of dividing cells to remember that state, or you just have this sort of unstable programming. So once you set those switches, at certain points in development, it's important for that memory to be maintained over a cell population. So those are some really cool things about epigenetics, and why people are, fascinated with how all of that works, right? How is all of that actually regulated by this whole slurry of proteins that are all packed inside the nucleus? So PCTF is a fusion protein, right? So this is an engineered protein, and our general approach to that was to sort of borrow pieces from pre-existing proteins where in cases where those pieces had very well defined and very well understood functions. And then so this is very consistent with how synthetic biologists do things in general. You go and you sort of look, search nature and, the literature and all the research for short stretches of DNA and protein subdomains that have very specific, well-characterized functions. And then you ask, okay, so given that this module, right, you kind of look at it like a Lego brick, and then ask if you could build, based on some general principles of, control theory, right, things like toggle switches and feedback loops, bistable states and all of that, can you put together well characterized biological modules to build something that behaves, right, like that mechanism that you would see in a machine? So yeah, so PCTF, again, polycomb transcription factor, that was the very first prototype for something that we that I wanted to build, where I basically, basically using the mechanisms that cells use to decode the biochemical information that helps to, establish like these epigenetic mechanisms. I wanted to build a protein that could actually sort of read those biochemical signals in a new way, and then its output would be changing the regulation state of a gene. So PCTF was built to read a biochemical signature that shows up on chromosomes. And then so the structure, or sort of the substance that we work with, is technically called chromatin, right? So chromatin is like this sort of, a compacted strand where DNA is wound up with proteins to help, pack all of that DNA into the nucleus. But that packaging is also sort of a control mechanism for controlling whether, or determining whether a gene is going to be expressed at really high levels versus low levels. So being able to control that, is potentially very powerful, and it's almost kind of a workaround. So when you can try to control gene regulation a couple of ways in cells. You can either take a piece of DNA and shove it in the cell and force the cell to express it at a certain level, or you can take the 20,000 different protein coding genes that a cell already has, and then just sort of nudge expression up or down in a very controlled way. And so that's what we do. So it's almost like, in situ, gene reprogramming. Since we published on that PCTF protein, since it was, as successful as it was, I started asking, okay, well, can I make this a general platform, right? So there's much more to control than just finding an off signal and turning a gene on, which is what we did with PCTF. We could go and find an on signal and turn down the expression of a protein, and that would be useful for, like, runaway expression of oncogenes in cancer, right? So and then we asked another important question, and this is the topic of a review that my grad student Kiera Franklin wrote. Well, if you then go out and try to identify all of the cell development diseases where this sort of epigenetic reading and output mechanism is messed up, what diseases are known to be linked to something like that? And we found things in the category of neurodegenerative disorders, right? Neurodevelopment disorder like Rett syndrome, autoimmune disorders like lupus, and as well as I think there's some examples just recently of cardiac diseases being linked to that mechanism, right? This epigenetic misregulation. So there's lots of potential, with us sort of using this epigenetic read and output engineering system to try to control and even just study, right, for basic research, epigenetically mediated cell regulation. So recently, we've actually started collaborating with Ed Bochway's lab to see how something like a PCTF, and then nowadays we actually call these synthetic reader actuators, or SRAs for short, so that we're not sort of locked into that, just focusing on one type of epigenetic signal and one type of output. We want to be able to mix and match and change gene expression in either direction in all sorts of different contexts. And then we've also started collaborating with Manu Platt, who's now at the NIH, and using some of this technology to see if we can influence how proteases are expressed in cells in a disease context.
That's really cool. That's a really interesting concept. We're nearing the end of our time, so
Okay.
we're gonna end with the question of what advice would you give to student researchers who are interested in similar topics or maybe even your lab?
Yeah. So the advice I would give is, well, first, if the question is, okay, well, what do I need to know, right, in order to, kind of feel comfortable, diving into research in the Haynes Lab, I guess my quick answer to that would be gene regulation, gene regulation, right? So even if you don't know much about it, if you're fascinated enough to to learn very quickly, then you're in good shape. If you feel like, you've learned a lot about it and you're still fascinated and you need to learn more, then that's great. So yeah, we're very sort of like, cell nucleus focused, right? Everything that's going on inside the nucleus of a cell, which is like, I guess generally regarded as like the brain, right? Or the genetic brain of the cell. Not no shade to the mitochondrial DNA, but we do focus on the nucleus. So sort of that sort of very central, foundational concept would provide a very good, I guess, sort of framework to make sure that that the person, when they're sort of confronting all of this new knowledge about chromatin and all the biochemical modifications to it, having a strong anchor and a strong grounding in just gene regulation and what the consequences are for gene regulation is very, very helpful. So that would be my top bit of advice. Also, I do want to mention that in any research lab, right, anyone who's considering working in any research lab in academia, working for a research lab is like working for like a small startup company, depending on, how young or old that lab is. The product is discovery, right? The product isn't how many hours did I clock today, or, did I check off these boxes on my, required, research track? While those, sort of checkbox things are very important, what's really going to ensure success and make your position fulfilling is discovery output, right? There's also been a lot of conversations around salary. Okay, well, my goal is to get paid X amount of money. That's fine and it's totally understandable. And hey, we've all got to eat. But I think it's I think what I've seen not get as much thought and consideration, I guess recently as is it, it's very important to consider the fact that when you're stepping into a research lab, the output is discovery, right? So this is why they say, oh, well, you need to be the type of person that can thrive in the face of the unknown, and sort of answering questions and solving puzzles. Well, that's why, right? And then so those products are in the form of presenting at conferences, presenting posters, writing papers. And that is the reason why, is that a lab can thrive and survive, and usually the PI themselves is that's their central motivation. That is the priority product, is discovery, right? Discovering new things that will hopefully materialize into the things that are useful for human health in biomedical engineering. Okay, so
Yeah,
that's the answer to that.
that's great. Yeah. I'm sure students are going to really take that to heart. Well, again, thank you so much for meeting with me, taking the time out of your busy schedule to come and answer these questions. So yeah.
Awesome.
One last question. Where could students find you if they have more questions?
Oh, yeah. So if students want to learn more about what we're doing in the lab, we have a website. So that's where you're going to find the most up-to-date information. Yes, so we have a WordPress page right now. It's K Haynes Lab, KHAYNESLAB.wordpress.com.
Okay. Sounds great. Again, thank you for taking the time to meet with me. That's a lot of great information. So yeah, thank you.
All right, awesome.